Started. And I want to welcome everybody to the start of our Global Genomics and Health Equity Seminar Series for the year. So excited to have Dr. Latrice Landry. Is it Landry or Landry?
Landry.
Landry speaking today. So I'm going to embarrass her by giving a really long intro. So Latrice is an instructor specializing in systems-based approaches to building equity and biomarker translation in clinical genomics. She received her master's degree in policy and her PhD in nutrition from Tufts University. Her doctoral research focused on the interactions between diet and genetics, studying dyslipidemia in African Americans in the Jackson Heart Study. She's got lots of awards. As a doctoral student, she was awarded the Albert Schweitzer Fellowship, nominated as a finalist in the American Society for Nutrition's Clinical Emerging Leaders Award, and was given the Presidential Award for Citizenship and Public Service at Tufts University. After that, in 2015, she joined Harvard Medical School's Biomedical Informatics Fellowship Program, where she has been studying biomedical information systems as tools for biomarker translation. In 2018, she graduated from the Harvard Medical School's Clinical Molecular Genetics Training Program, enabling her to integrate her biomarker experience with direct patient care. She was the inaugural Food and Drug Administration's Genomic Medicine and Minority Health Fellow, and has also been recognized as a thought leader in minority health and precision medicine by the National Minority Quality Forum in 2017, as a top 10 under 40 rising star in business and academia by Genetic Engineering and Biotechnology, and in 2018, and as an American Society of Human Genetics scholar, human genetics scholar in 2019. I'm just going to stop embarrassing you now and just let you go on and begin your presentation. And as I explained to Latrice, we try to keep this informal and we really encourage discussion. It's also an opportunity for people to raise new ideas about, encourage opportunities for collaboration and so on. So please take it from here, Latrice.
Absolutely. So thank you for that introduction, Sarah, and thank you all for this warm welcome at Penn. I will say that I took Sarah's indication to me when she invited me to speak today to keep it informal, to even sort of present ideas and sort of use this as a platform for building collaborations on intended projects and future things. So I'll first just start off a little bit with just a little bit of my background. So I usually like to start by introducing myself outside of the accolades and sort of the formal academic self that we document on paper. I began my journey in this space of health equity research in high school when I decided, despite my love for the theater arts, that I would pursue a career that would help me sort of think about and evaluate and hopefully eliminate the disparities in my community. I grew up in Prince George's County, Maryland. And I think one of the things that always impacted me, that it just seemed inevitable at a certain age that you would experience diabetes, heart disease, cancer, sarcoidosis, and other sort of things that were plaguing my community and my family. And so what I thought at the time was that you go and study medicine, you become a doctor. That's what you do when you're interested in health. And so I went on that path. I discovered the field of community health at Tufts University, where I went to undergrad, and fell in love with this idea of research and really understanding social determinants of health and combining that with some of biological determinants of health to really sort of impact health outcomes, and not just individuals and community, but communities. So that pivoted me away from individual medicine to more community-based medicine. From that perspective, I then went on to pursue the doctoral degree that Sarah mentioned in gene-nutrient interactions, this idea of balancing nature and nurture. And that's sort of where I've been, and really sort of thinking about how who we are impacts interacts with the environment that we live in, and that impacts our health. And from there, I went on to really sort of thinking about how we translate this into actual practice and research and actual clinical care. And that's led me to the space of thinking about health systems. So I like to give that because I know that my introduction often meanders and it gives a sort of variety of different fields. But I've truly come to a space where, again, I started with this intention of how do I really focus on eliminating health disparities and understanding health equity. That led me on a journey of a variety of different fields and disciplines to ultimately be in this space where I'm really looking at how we change the system, right? And so one of the things and one of the systems that I'm looking to impact is this system of clinical genetics and how we translate and how we translate clinical genetics from bench to bedside. I'm a laboratory geneticist, and so I think about interpretation. So that's where my talk is going to start today, really thinking about the pipelines in clinical genomics, how we interpret variants, and what are the needs we need to do to interpret variants more equitably. So give me 1 second. I think you should be able to see my slides. We'll look for a shake of the head to.
Yeah, not yet. There we go. They're coming on.
Perfect.
Now, the only thing is we're seeing the notes on this.
Oh, that's interesting. Okay, let me.
I know that's happened before, and I'm trying to, do you have to do like the swap the display or something like that?
It was next slide we were seeing, yeah.
Okay, let's see, I'll do it again. And you said swap the display?
Try do the swap displays. I think that's worked before. That's it.
Perfect.
Yep.
All right. So it's sort of to highlight sort of this sort of space we're in, and what I would like to talk to you guys all about today is a recent RFA from NIH, which is on the use of multi-omics and incorporating that into understanding health and disease. From there, I focused on thinking about multi-omics as really one of the ways in which we might think about interpreting or reducing the disparities in diagnostic yield for variants of uncertain significance. So the title of this sort of brainstorming session around this is the multi-omics approaches to optimize interpretations of variants in minoritized populations. And really, we're talking about VUS variants that you guys are all very familiar with, which is one of the classifications of a variant in a genetic assay that leads to uncertainty. Essentially, we don't know if it's pathogenic, we don't know if it's not, if it's benign, and we don't know, we don't have enough information or evidence base to move it in either direction. Just to give a little bit of background, give me a second. It doesn't seem to want to advance.
Now we're seeing a doc there. Let's see if you touch on the presentation and try it, it doesn't do it.
Yeah. I apologize for this.
Is there a little arrow? Is there a little arrow in the lower left that you can use? Put your.
Oh, I see. Thank you, Dan, for that.
Try that.
Okay.
Yeah.
Okay. Thank you, Dan. Okay. All right. So, one of the things I like to start with is sort of when I started and really started thinking about the ways in which we do laboratory medicine. I actually started my laboratory medicine training and completed informatics training at the nascence of the Precision Medicine Initiative. So here is a slide. This was a very exciting time for me. So this is then President Barack Obama, the Precision Medicine Initiative with Eric Green's graduate degree, DNA double helix. He's very proud of that. He mentioned that in an informal interview I had with him, that this was actually his from graduate school, used as a prop to announce the initiative. And so part of the excitement about that is that the Precision Medicine Initiative is meant to transform medicine as someone who was completing an informatics degree, I truly believed in that possibility of transforming medicine with use of biomarkers. And here I was beginning on my my laboratory diagnostics training as a FDA-funded inaugural precision medicine and minority health fellow. So the task I was given is to see and understand how this laboratory genetics in the field of laboratory genetics could be used or does impact minority health. What are any issues and what are the things that could be highlighted for the FDA's Office of Minority Health within this initiative? It's a very broad but big, a lofty goal, and I was an N of one. So the first thing we started to talk about was to break up this. I apologize, it looks like it's sort of reformatted my slides a little bit, but just bear with me. So the first thing is that at the time, the FDA and NIH leadership councils had sort of stated or defined classifications of biomarkers, right? And so part of this and part of the Precision Medicine Initiative was on biomarkers. They decided they needed to define the classifications of biomarkers. The first classification was predictive. And so the first biomarker classification is predictive of treatment. So these are the categories: is predictive of treatment. They could be also diagnostic biomarkers, so they'll help to diagnose disease. They could be prognostic biomarkers, meaning they could sort of inform the prognosis over time, and risk-based biomarkers. These were the four main biomarkers that impact more genomics and sort of rare disease. But they also have others like disease monitoring, which are very common in cancer, and sort of safety and efficacy biomarkers that were sort of outside of the purview of what we were doing mostly in laboratory genetics. But these were the categories of biomarkers we were most concerned about. You can imagine predictive of treatment. We have lots of classifications of that, or lots of examples of that now. Diagnostic is mostly what we were focusing on in the context of my training and research around this area. But risk is really sort of what I suggested they focus on in the context of polygenic risk scores going forward, primarily because of their influence by population differences. So when we're thinking about this, primarily we just started focusing on sort of really sort of helping the FDA sort of understand the role of laboratory genetics and in precision medicine. And so that's starting with identifying sample types. I do some of this, or I'm presenting some of this just as a background, sort of thinking about the multi-omics approach that I'd like to discuss. And sort of looking at biomarkers. Now, traditionally, in our genomics labs, a lot of the biomarker, sort of the biomarker of choice is usually a DNA-based biomarker. But we know that there's lots of other biomarker types. So partially what we suggested to the FDA is that that the template for how we think about biomarkers be rooted in DNA, but that we could expand that template to impact other biomarker types, including RNA, proteins, metabolomics, and sort of even thinking about the larger microbiome, now classified as the pangenome. And then thinking about those biomarkers, really to sort of in this space, think about the different classifications of biomarkers. So these are our test results. And so this is the framework we set apart for the FDA Office of Minority Health, and how we could think about biomarkers, thinking about the individual marker, the actual specimen type, and the results, and the ways in which we could improve it. Now, for the multi-omics RFA, the idea here is that DNA is required. I don't know if you guys have seen it. I will pull up the RFA in a second. But the DNA is required. It's a required component of your proposal. And then you have to select up to 2 other biomarker types that could be included. They could also be DNA-based biomarkers like epigenetic markers, as well as they even have a sort of an option to include, in addition to sort of coding sequences, you could sort of think about in-depth measures to capture non-coding sequences and capture that or frame that as a multi-omics approach. All right, so when we're thinking about this in terms of the overall broad-based genetic testing report, what I'm proposing and what the RFA calls for is inclusion of multi-omics, but it doesn't call for it to be focused on clinical genetics. this is sort of my application for it. So including multi-omics to sort of think about the molecular diagnostic findings. These were the categories put for us: causative agents, stochastic modeling. So could we use multi-omics to build stochastic models for predicting disease, improving therapeutic prospects, and disease treatment? My intention or focus is really focused on the diagnostic aspects. To think about it now, recently they sort of reframed this in sort of looking at less of a linear approach. They're looking at this in terms of a cyclic approach, or sort of a wheel of biomarkers where things can overlap. Here we can think about the idea of the multi-omics, and especially depending on certain disease applications, you have this sort of this cyclic nature of the diagnostic and the predictive, meaning predictive of treatment, being on a continuum. Well, often we might diagnose or even think about prognosis by identification of a driver, for instance, in the case of cancer. And then that driver might also have a targeted discovery. Okay, so here's the RFA. And again, I just wanted to use this as an open discussion, as Sarah had suggested, because this has a rather quick turnaround. So I'll walk you through sort of the original thing of when I was first asked and what I was thinking about, since narrowed down a little bit. But I wanted to sort of highlight the RFA. So essentially the RFA is asking for general thoughts about multi-omics, which has been a long sort of area of focus for NIH, and thinking about how it can impact health and disease broadly. Again, I would like to focus that on clinical genomics and focusing on the use of multi-omics and the interpretations of VUSs, which are at a higher frequency and minoritized populations. So I've thought about this, and some of this is just sort of the framing of this as an orthogonal variant interpretation approach. Now, some of you who are in this space of clinical genomics will remember the orthogonal variant interpretation approach, or orthogonal genomics, as an approach that was presented by a diagnostic laboratory called Claritas, and which no longer exists, but essentially they use two individual NGS instruments, one the Roche instruments and also Illumina, to triangulate and improve the validity of the NGS variant calling. They called it an orthogonal approach. So using two different approaches, they could look at the discordance and concordance between the two approaches. And it was a very expensive approach to identifying variants, identifying pathogenic variants. But they had essentially been gifted machines, and so they could run that approach. They're no longer with us, and I think that it was a very expensive approach that wasn't really replicated in other places. But the idea that you can use different modalities to help triangulate and identify and interpret variants is actually a valid one. So they used it in the concept of validating the individual DNA or nucleic acids that were identified as varying and potentially pathogenic. In this case, I'm not really thinking about validation. We're thinking about clinical utility, and also the and focusing on the interpretation of variants. So for this, approach, what we would do is sort of first identify a disease cohort. NIH asked for a small sample of 300 individuals, which 200 are disease and 100 controls. And the idea here was that we could identify different multi-omic patterns, and then use that pattern and then see how VUS patients who have already been diagnosed with or sort of have a test result of a VUS segregate with either the disease or the controlled patterns. So essentially we build the multi-omics pattern with a cohort of disease and those who have controls in a training test set. And then we look at our VUS participants and see how they segregate within within the larger diseased and or within the larger training and test set. Sorry.
Latrice, I have a clarification question. You maybe you're going to say this in a moment, but I'm still trying to understand, is your target, are you going based on a disease, a clinical, like, I don't know, hypertension or whatever?
Mm-hmm.
Or is it focusing on people who've been identified because they have a variant of unknown significance? I guess I'm thinking of like Mendelian versus complex traits and.
Yeah, so Sarah, that's a great question. So when I started this, when I first was thinking about this, this was actually something that I had suggested a while back. However, you need to have a proof of concept. So you have to have a disease in which you can showcase that using multi-omics can help improve interpretation of variants, right? And so if you're familiar with the clinical variant interpretation pipeline, we often use in silico prediction models, other things we pull into the actual interpretation process that help us sort of think about things and get to a more definitive answer. So for this one, I have to arbitrarily choose, and I say it arbitrarily, but there's some thought into the disease model. One of the disease models that I have, and I can actually just jump ahead a little bit, but ultimately, I think the disease that presents itself best, because I'm doing a little some other work with it, is cystic fibrosis. So Sarah, for your point, the idea here is to use cystic fibrosis as a disease model by which we can showcase that using multi-omics or integrating multi-omics approaches into the variant interpretation pipeline can improve or decrease both the VUSs, but improve diagnostic interpretation for minoritized populations. So for this disease, when I generated this slide a few months back, it was focused on just disease and controls, and we were going to select, sorry, we were going to select a disease. And I actually thought that we'd be able to do that in this group, just talk about it, because I had started preparing research on FH. I have a cohort that's focused on COVID that has over 3,700 individuals in it. Was thinking about doing something complex like that. But because the RFA has such a quick turnaround, I really needed to focus on a disease model. I'm a quick writer, so if people, I want to sort of think through this with the group, because the RFA has some very specific requirements. And so if cystic fibrosis doesn't seem like a great idea, I talked to a few other people, including Håkan Håkansson at CHOP. He was very supportive of using cystic fibrosis and thought that this would be a great model for this type of thing. But that's kind of what I want to discuss. I also want to discuss the specific components. So the larger idea is that we'd have patients who were diagnosed with cystic fibrosis with pathogenic variants, right? So the idea is to be diagnosed, you have to be identified with you have to have a pathogenic variant or a likely pathogenic variant on your report. And then you'd have controls. And we would look at the patterns and select the other multi-omics modalities. Cystic fibrosis specifically lends itself to proteomic approaches. There's a lot of research on proteomic differences with patients who have cystic fibrosis, so really looking at that. Some of that data is specific to sort of looking at saliva secretions and sort of nasopharyngeal secretions, so they wouldn't be, so that would be the specimen needed for that. So partners who have access to that. But again, I mentioned that I have talked to some people who have access to, CHOP has a cystic fibrosis cohort in clinic. Doing some other work with CHOP, have access to data of patients and variants. So we would select variant patients who had VUSs and sort of the non-white patients who had VUSs. And so all of that is very doable with their database, and as well as access to patients for control. Again, it's a small, so what NIH is looking at is for a pilot. What they're also looking for, they're building a consortium. So they're not looking for, each consortium will have its own diseases. The submitting institution would be NHGRI in this case. NIEHS and NCI wouldn't be appropriate for a cystic fibrosis application, that was something that I had explored with others. And the idea here is that they aren't sure if you're going to get all of your multi-omics approaches that you want. And so what they're looking at as sort of the types of multi-omics assays that you propose, your ability to recruit participants for this study, and collect new data, and sort of building both pipelines. There's going to be a individual sites, and this is what this application is for, is a site to do this. But there's also, in this case, for the RFA, in addition to the sites that they have available, they're also funding a sort of computational core, as well as a multi-omics assay generation core. So I do know that this is falling into, and speaking with some of the program officers, this falls into their exploratory RFAs, where they're saying, show us that you can do it, and we're going to figure out the nuts and bolts later, but give us a plan of what you would like to do. So that's where this fits into. And essentially the idea here is then using just a clinical genomics application. So we have
Be careful.
Oh, go ahead.
They may just need to mute.
Okay. So I think what I had planned to do is that we discuss it. So one of the things that I had thought, and in the letter of intent had kept it open, and they said that that was fine, but it does need to be submitted at the end of the month. So cystic fibrosis has allowed me to start to build out the individual components. And I think one of the things that you had said, Sarah, is that, through the audience, that there's lots of people who are interested in lots of different things. I have, through my short time at Penn, built some connections with individuals in the Department of Genetics. I work with Marilyn Ritchie. I work with Halkin Connerson. But, sort of one of the things in this case is sort of thinking about are there others at Penn and or CHOP who have interest in this space and could be collaborators on a proposal like this?
That's great. Can I ask some more clarification questions?
Absolutely.
Do you have more slides to show to discuss it?
I do, but they're really sort of building the case for cystic fibrosis. And I think that sort of going through the data and the disparities might not be the best use of time. So I really would like to sort of get more feedback. So we can pause there if you'd like.
Well, I noticed that one of the emphases is on environmental, so social determinants of health. So how would you propose to collect that for the cystic fibrosis patients?
Right. So for the, there are 3 partnering institutions. And so NIEHS has a strong interest in social determinants of health and environmental assay collection. However, it's not a requirement for all of them. So it depends on where you're submitting to. This is what they, the feedback that I got from them, but also what they presented on the webinar. And so essentially NIEHS has a strong interest in that. They do encourage those who are not submitting to NIEHS, NHGRI or NCI to explore social determinants of health where appropriate, but it's not a requirement for the other branches.
Okay.
And so, but I will say social determinants of health is of interest of mine. I think for this particular model, what I was trying to do and go through were, so one of the things they wanted to make sure is that people would present with disease in the 5-year timeline of the grant, right? And so, certain diseases, cancer was one of them, although NCI is interested in it. For NCI, they were thinking more of patients who already had cancer and thinking about things like reoccurrence. And so things that would be more likely to happen a 5-year time than waiting for instance, a cancer outcome or first occurrence. And so they're really open, but I do think that if, in talking to Halkin, there are environmental things in general for all of the respiratory conditions. Cystic fibrosis does lend itself well to, for instance, nutritional observations or some.
Careful, be careful.
Can I just ask everybody, please mute if you're not speaking? Thank you. Sorry.
No problem. So yeah, so cystic fibrosis does lend itself to nutritional exposures, which do classify under environmental exposures. There's a lot of work on cystic fibrosis and vitamin D. If, for instance, we wanted to do that, in which case collection of vitamin D would be quite easy. I even was trying to identify individuals at Penn who would sort of do vitamin D, and it seems that there might be a number of potential labs at Penn who could do that. And so the main thing is the genomics, and that's a required component. The options, I also say that the assays and the funding of the assays is not a requirement for this RFA. That would be under the center. So you don't have to budget sequencing, transcriptomics, or anything like that. That is focused on the data.
I can show you. No, it's fine.
Sorry, there we go.
So yeah, so and then the big focus is on recruitment. And so you know, sort of was putting this together and sort of going through sort of cystic fibrosis is interesting because it also has so many different phenotypic, so many different ways in which it is phenotypically expressed. But one of the things we could think about in terms of environment would be nutrition. I think it's the easiest. In speaking with Halkin and CHOP, they do have environmental measures that in ways in which they collect data for CHOP for other studies like asthma. I don't think that would be the best approach for this RFA for cystic fibrosis.
Could I, again, I'm sorry, others should jump in, by the way. We want to hear from lots of people if you have ideas. I'm hoping for some more clarification. So cystic fibrosis, of course, is it's most common in people of European ancestry, but for that reason, it's often been ignored in people of other ancestries. And so I could see why this would be important. And I guess what I'm wondering is, in people who don't self-identify, let's say, or based on genetically inferred have European ancestry, are they known then to have, do they have more often these variants of unknown significance? I know there's like one really common, that Delta mutation.
Delta 508, yeah.
Right,
Yep.
But I'm taking it that in other ancestry groups that there are other variants of unknown significance.
That's exactly right. I actually had a slide prepared for that. But just to just talk through it, because I don't think the actual numbers are as useful in this context. But yes, you're exactly right. So cystic fibrosis has been generally studied and well characterized in populations of European ancestry. However, the last 10 years, there's been increasing data suggesting that it is underdiagnosed and patients of non-European ancestry, with the largest sort of research endeavor going on in California at Stanford, trying to characterize cystic fibrosis variants in Latino and Mexican populations. The issue here is that, yes, Delta 508 is still commonly thought to be the most common cystic fibrosis variant, disease-causing variant. However, it only captures, even in Europeans, it only captures, maybe 70% of cystic fibrosis patients in European populations have one Delta 508 allele, and most will have other allele, and it decreases in each ancestry. So for instance, for Africans, for Asians, and for Latinos and Native Americans, the prevalence of Delta 508 is less than 50%. And so when we think about ACMG has a standard sort of 23 variant panel that it suggests. But when you look at the diagnostic yield of that specific ACMG panel, it performs at about 90% for patients of European ancestry, and that decreases to 49% for patients of Asian ancestry, 60% for patients of African-American ancestry, and about 70% for patients who identify as Latino or Hispanic. So the actual test yield, the diagnostic yield decreases because the variants that have been identified and characterized and therefore have an evidence base that says they're pathogenic is much lower for non-European populations. And you know, so it's suffering from that. Again, this is the idea that we can use multi-omics to improve. And so in some ways, having a disease that's well characterized and having even potential access rather to a patient population where we can have a lot of phenotypic data on them through maybe the CHOP clinic is something that is worth considering. The other thing to think about cystic fibrosis, and this is specific to maybe pulling cystic fibrosis patients out or identifying them in the Penn Biobank, is that certain variants lead to, or classifications of variants will lead to more severe disease than others. And so in minoritized populations, it's currently believed that there are higher rates of undiagnosed individuals with cystic fibrosis. And there's even an organization in Pennsylvania, an African-American man who was diagnosed with cystic fibrosis in his 40s, so in his adulthood, after having years of being sort of sent away with pneumonia, bronchitis, asthma, different sort of diagnoses that just kept being recurring and kept persisting into adult age. So finally he got sequenced and identified that he had cystic fibrosis. And he and his wife have started an organization because the data suggests that minoritized populations are more likely to be undiagnosed in childhood. So to have those undiagnosed adults is more common in minoritized populations because they were less diagnosed as children.
But that would also, I would assume, also have to do with partly access to healthcare, and then the fact that people are not looking for it because they think, oh, this is only in people of European ancestry, but they might still have a common variant that's already known. But here you want to focus on people who have
VUSs, right? So that's because of the multi-omics approach, right? So for instance, you could have, if you wanted to sort of think about improving diagnostic yield of cystic fibrosis and minority patients, you could just say, I think my hypothesis is they're just not getting tested as children. They're going to have a common allele most likely. And so I want to just make sure that increase the numbers of minoritized populations that are getting access to testing at certain time points. The reality is, though, Sarah, and you're probably an expert at this, right? So you know this well, that there are more private variants, there are more variation in cystic fibrosis variants in African populations. Our African ancestry populations is the highest, but there are particularly more, so for instance, of the top variants of each population, most of them are captured on standard assays in European populations, but you get down to less than 10% in African populations, the most frequent variants. And then with African ancestral populations, you also have a high number of private variants that are potentially pathogenic, right? And so those aren't being captured. And you may have been thinking also there's standard time points in which people are being reviewed for cystic fibrosis or screened for cystic fibrosis. But even in newborn screening, it's not regulated across states. So for instance, the state of New Jersey only tests for Delta 508 in newborn screening. And so every child who doesn't have Delta 508 would then have a negative test. So it's a combination of things. And so this RFA is specifically saying how can we use multi-omics approaches in health and disease. And so the point is that I believe by using multi-omics to help sort of help us understand the individual pathogenicity of the variants in this database that are identified as VUS, we could improve variant interpretation and diagnostic yield for those patients. But you're right, Sarah, there's always going to be the issue of access, but that is a separate issue.
So, and what are the different types of omics data? You mentioned proteomics. Is there something else that would be informative?
Right. So for cystic fibrosis, there's a lot of data that suggests that, or a few studies that are sort of highlight metabolomics. So that's another omics platform that is of interest. And then again, the NHGRI has said that they are also open to use of, for instance, non-coding regions. So for instance, a lot of what's focused on is exon coding. So you could say that by targeting non-coding regions or using a whole genome versus exome approach, that is also multi-omics. So yeah, so those are the three that are coming through in the literature. And that's important because although the RFA is exploratory, they do want, you know, a suggestion that there is likely, or some evidence that is likely to impact the outcomes that you're suggesting that it will.
Okay. I would love to encourage people in the audience, if you have any suggestions, and if you do have some other slides that you think might be helpful, please feel free.
See if I can pull up let me stop screen share and just go straight to the slide where you were asking about the variants, because I think that might be helpful to see. I think also the issue I have is that one of the things that I think is useful in using rare disease as sort of a pilot for what we'd like to do in clinical genomics before expanding to more complex disease is that clinical genomics is rooted in rare disease. A lot of the stuff is more established. I actually trained in clinical genomics after coming from a background in population genetics, sort of working on cohort data in the Jackson Heart study, primarily because the practices and the rules and the guidelines for clinical genomics are rooted in rare disease. And so piloting something in rare disease and then building on it for complex disease, I think is a strong case because there's, you know, in terms of clinical implementation, there's a lot of things that you need to do. I see Arupa.
Yes, I would like to hear Rupa's feedback.
Yeah, I have some.
Rupa, you're muted. Somehow you just got muted. There we go.
Okay, hi. So Latrice, yes, I've been following your presentation, but I have a throat issue. That's why I hadn't unmuted myself before. My question is really, you know, in terms of your proposal, you are talking about what Marilyn has done and what Hakan has. So those are data sets that's available to you. And in that population, what's the representation of the minority population? And in that population, what's the fraction that's VUSs? As you know, that at the ASAG meeting, there was a lot of discussion about the VUSs and the evidence-based medicine that would put one in one category or the other. And what I'm curious to know is that when you are talking about multi-omics, whether, you know, you're looking at RNA allele specific expression or at the level of proteomics, some difference, what would be your cell line that you would use, or is that data already available, or as you are saying that you'll be generating the data? So from what I hear, it seems to me that it's a very, I mean, in theory, yes, it can be done, but really, what are the things that you need to have to make it work? Can you give me a little bit more details on that?
Yeah, so I think one of the points here is that the way the RFA is written is that you're essentially, or the part of the RFA that we're responding to, or the specific RFA that I would like to respond to, is focused on a site for a specific disease. So you develop the site around the specific disease. Because of that, and because there's going to be a multi-omics core, you don't have to generate, right? So, and they also don't want to. So for instance, I can't say I'd like to, we have, you know, state-of-the-art here at Penn, we're going to do it ourselves, a Penn application for being the course. They want it to be comparative, even though we might have different diseases. So for instance, I could put in an RFA for a cystic fibrosis. Someone else could put in an RFA for breast cancer. Someone else, but they want all of the assays to be done on the same instruments from the same core or from a collection of cores. So for instance,
So it's like a centralized core.
exactly, similar to like Emerge or some of a few other sort of multi-site proposals that have gone forth. And so essentially what I would need is to, one, have access to patients and sort of the controls as outlined. And Arupa, your question about the number of VUSs, as well as the number of minoritized patients, for instance, in the two different biobanks that we could identify, is a very valid one, because that's essentially the limitation. You have to prove that you have access and that you could identify these individuals, and then you have to have a recruitment strategy for recruiting them. For the CHOP biobank, what my understanding is, and we're trying to still pull those numbers, we don't have them by VUS. But for minority populations, essentially non-white, we have two ways to do that. And so one of the ways is we can look at Ancestry, because they have genotyped much of their database. And so by looking at Ancestry, and depending on how we, the way it was put to me, because I haven't actually, they're pulling the data for me now, but the way it's put to me is that if you look at Ancestry and you define European ancestry as no non-European ancestry globally, then you have a very large data set to work with. I'm not sure that I want to approach it that way. So to define non-European ancestry so diversely, because I think that winds up losing the impact, right? It kind of sort of waters it down. And so we need to look at the numbers. But by EHR classification, I'm told over 30% are non-white for their cystic fibrosis registry, and that's just CHOP. And for Penn, it's slightly larger. And for Penn, we're actually, there's an in silico model trying to pull out cystic fibrosis. So not looking at the diagnostic code, but looking at the EHR records. To identify undiagnosed cystic fibrosis in the Penn biobank. And so we're still trying to get that cohort so that we can actually pull the numbers. But the thought was that there'd be more undiagnosed minorities in the adult biobank. And so perhaps that number would be larger than 30%, but that is the exact answer that we need to get to ASAP, which is kind of why I had to sort of like pivot and just pick a disease model where I thought we could actually get cases, because trying to drill down some of the numbers in that work needs to be the focus to get it out on time.
So Latrice, I would recommend focusing on particular ethnic groups or ancestries, because they want control. They want like disease and control. And if the definition is just not European ancestry, that could be so diverse. And with those small numbers, you're only going to have 100 controls and 200 cases. So try as much as possible, I think, to define, and that's going to depend on what's in the database, certainly. But Arupa, I thought you were also asking, like, what cell type are you going to do?
Yes. Yeah,
the multi-omics.
That was indeed my question, is that what do you need to do the omics analysis? Do you have to have access to, I know Penn Biobank has lymphoblast cell lines for all of them. Are you going to propose that based on this, you are going to generate iPSCs and then look at the consequence of the boost? Or are you going to think that in Hawkins' database, they probably have collected some of the proteomics? I just think that at the end, when you pair up the cases and the controls, and then you pair up the boosters, which are, I mean, you don't know whether they really have any consequence or not. The power of numbers can become very limiting in that particular omics data set. So, but I'm sure since they're calling it exploratory, they will accept a lot of leeway in that analysis. But I just think that, how will you identify the cases and the controls, and how will you weed out that they have no real disease at all. That's the thing. And cystic fibrosis being a recessive disorder, you know, that's another thing. What will you do with the heterozygotes, and what will happen in terms of their omics?
Yeah, so I think Dan had once asked about haploinsufficiency in cystic fibrosis. I think that partially here, essentially we're getting for a numbers game. And I just talked to Halkin on Friday. And so I think I should have more data on the numbers maybe tomorrow or early this week. But essentially it's sort of playing that game between having the most robust informative data. So for instance, if there is a possibility in CHOP, I don't think it's the same at in the Penn Biobank of having sort of actual a lot of the proteomics that is of interest is from saliva or sort of nasopharyngeal swabs, where they're sort of collecting some of this data for certain types of assays. I don't think I need to sort of follow up, but Dan, if you have an answer, that would be available at the Penn Biobank. It is, but at the CHOP Biobank, primarily because they had the cystic fibrosis clinic, so they've collected specimens over time. Part of what we're trying to do is then see, hey, how many of the specimens you have are in patients who identify as non-white based on EHR classification. And so it really is trying to think about a numbers game. Ultimately, though, for the RFA, I don't actually have to propose a specific cell line or anything like that. If I think that DNA and blood is all I'll have access to, then I can propose. There are some suggestions and some strong papers around sort of epigenetic effects of some of the variants. And so I could propose it, avoiding some of those more technical things that I think that I won't have access to or may be difficult to collect.
Thank you.
Yeah, I think that thinking about the cell type is going to be important, because you could get anything from lung biopsies, you know,
Right.
to, as you're saying, the saliva or the nasal swabs. And I don't know the literature enough to know if blood it can be used as a biomarker. I don't know, Dan, maybe you have some information about that.
No, not necessarily, no.
Yeah, so for this RFA, they say the requirement is genomics. But if I were to say, for instance, we're going to use PacBio, we're going to do hi-fi reads, we're just going to do genome sequencing, and we're going to get the methylated DNA, then that's 2. And so that's required to have at least 2, the genomics and then the methylated. Sarah?
Yeah, but the issue is that the question of epigenetics is indeed interesting, but I don't know that blood, you know, what you're going to get from blood would be the right cell types to look at epigenetic factors influencing cystic fibrosis. So I don't know, others may know this better, but that would be my concern.
Right. So I get what you're talking about in terms of the actual comparison of diseased and non-diseased individuals. So yeah, sorry. Now I understand your question. I apologize for that. For specific cell types, I actually was looking at, so one of the, let me see if I can pull it up. One of the recent, actually, I think I'm having trouble finding it. But yes, so that was one of the things I think Halkin and I had discussed on Friday. And so I think that one of the things right now is we're trying to see what is possible. So I haven't sort of drilled down on that one specific aspect of it, but it will be based on sort of the numbers and what they think that they can have. But the benefit that we have is that the RFA doesn't require you to have, oh, I see a,
Yeah, I see a hand, Janaea.
So one of the things you can propose in future phases, so they said for a phased approach so that you could identify or collect that later. Janaea, I see your hand up.
Hi, yes. My name is Janae. I'm a 4th-year PhD candidate now in the Genomics and Comp Bio program, PhD. I was just wondering, since I also attended ASHG, a lot of the themes were surrounding kind of like race categorizations, further considerations for certain ancestry groups, how we group people, but also best practices for data stewardship, especially in large consortium in like multi-omic analysis, particularly with studies that affect populations that are minoritized. I guess my main question is, what's your purview of how we can improve that sort of ecosystem here at Penn, especially since we have such a rich resource that is, it's not UK Biobank, but we still have kind of a large data set that is pretty unique, especially to the Philadelphia population. So I'm wondering, just generally, even if it's not, related to cystic fibrosis specifically, just what are your general thoughts on that?
Yeah, I think that's a valid point, and part of the reason and part of the sort of exploration around this in this RFA is that because of the diversity in the Penn patient population and CHOP patient population in the Philadelphia area, we have an opportunity to add to a space in really any disease area that is essentially in clinical genomics. Sort of diverse data is largely unrepresented, and especially in African Americans or African ancestral populations. So I think that there's a value add. And so I think that's point one. And we could really have focused on any disease area. I was really just trying to streamline based on sort of access and sort of collaborators. But one of the key things here to your sort of question about data integrity and sort of thinking about things from that standpoint. The thing about this RFA that troubles me less, right? So when we think about large consortiums, a lot of times we're thinking about sort of open sharing of whole genome data of populations that have a lot of mistrust. And so there's a lot of concern about how you do that. What does broad-based consent mean? Are we doing that in the most responsible way? Or are we sort of opening ourselves up for a lot of harm by doing broad-based at the same, sort of changing the rules around sort of research participation, where now you give right, not just to a specific aim, but now we have access and are sort of codifying that in an agreement to your DNA to use for every, for lots of things, and including giving it to other people, right? So that's kind of the issues. And a lot of minority populations or vulnerable populations are very concerned about that. This consortium, although per the NIH data sharing agreements, will be shared, isn't necessarily the same, right? So we have a small sample, 200 of diseased individuals and 100 controls, right? So it's a very relatively small sample. They're going to be deeply sort of phenotyped and sort of genotyped with these multi-omics assays to go with it. But you're right, they'd still need the protections. My understanding is part of the protection aspect of it will be built into the data science core. And so NIH has been pushing to have all data on their platform Anvil. So rather than you downloading the data, you have to log into the data so they can control a little bit about where the data goes. So I don't know if that goes to your specific question there. And then just go ahead.
I was just going to say, we are at 4 o'clock now, and I want to thank everybody for attending. We can just hang on for a couple more minutes for those who are available, but thanks everybody. We're glad to be able to brainstorm these ideas in this format. But I was going to say, Latrice, that again, looking at the RFA, there was specifically an emphasis on community outreach, and that's what you could do that could be unique with only 300 individuals. You really could reach out individually to people and make sure this is done with true partnership and consent and sort of a new model for doing that kind of research. I think that would make you stand out.
Okay, I love that, Sarah. Thank you for that. And Janaea, if you want, I am going to put my contact information in the chat.
Sure, yeah.
Just so happy to talk offline. And I just wanted to thank everyone as we're sort of logging off. This is early stage. It was an RFA. Someone said you should apply because they knew some of the things that I had proposed early on. So literally early stage brainstorming. And they said, well, you're pretty quick grant writers together. So we'll see. It's going to be a true test of that. And I will follow up with everyone with the final product.